In this video, we're going to talk about the plasma membrane and how the plasma membrane interacts with both the cell contents and the cell's external environment. Let's start off talking about the composition of the plasma membrane. The plasma membrane is a double layer of phospholipid molecules with the polar hydrophilic head oriented both on the outer surface of the cell and also inside the cell, and then the hydrophobic lipid tails are oriented inward. So this is a phospholipid bilayer. And I think it's worth mentioning that there are many different types of phospholipids, including phosphatidylcholine, which is also known as lecithin, phosphatidylserine, and there's phosphatidylinositol. Now, that phospholipid bilayer is the basic structure of the plasma membrane, but it's not the whole story. The plasma membrane is a very complex structure, and it contains a lot of other things, such as a huge variety of proteins, such as transport proteins, tyrosine kinase receptors, G protein-linked receptors, and so on. By mass, there's actually more protein in the plasma membrane than there are phospholipids. You also have glycoproteins, glycolipids, sphingolipids like sphingomyelin, and cholesterol, which makes up about 15% of the plasma membrane in our cells. And cholesterol is a more rigid structure than phospholipids, so including cholesterol in the plasma membrane prevents the membrane from being too fluid. So a little bit of cholesterol stabilizes the plasma membrane, but if there's too much cholesterol in the plasma membrane, the membrane becomes too rigid. And additional cholesterol also increases the membrane's melting temperature. Now, one of the things that makes each cell type unique is the unique type and number of receptors that are incorporated into the cell's plasma membrane. And there are three main families of receptors that we're going to focus on throughout Part 2 of the course. There are tyrosine kinase receptors, G protein-linked receptors, and steroid receptors. Now, the first two types, the tyrosine kinase receptors and G protein-coupled receptors, are transmembrane receptors. They're part of the plasma membrane, and they're kind of embedded in the lipid bilayer, and that's where they perform their functions. But steroid receptors are found inside the cell, kind of floating around in the cytoplasm. Steroid hormones like estrogen or testosterone are able to pass through the plasma membrane, and then they bind to their receptors inside the cell itself. But tyrosine kinase receptors and the G protein receptors are located on the plasma membrane. Now, we're going to talk about the G protein receptors in detail in the Pharm Basics section of the course. But for now, let's focus on the tyrosine kinase receptors. Take a look in your study guide. Tyrosine kinase receptors are transmembrane receptors that bind to some extracellular ligand, and then on the part of the receptor that's inside the cell, they transfer phosphate groups to selected tyrosine side chains on specific cellular proteins. That's called phosphorylation. So a kinase is an enzyme that phosphorylates things. It just means it adds phosphate groups to things. And a tyrosine kinase only phosphorylates tyrosine residues. And the very first step in this signaling cascade is autophosphorylation, which means that the tyrosine kinase phosphorylates itself. We're going to come back to that in a moment. Some tyrosine kinase receptors, like the receptor for platelet-derived growth factor, are single-pass transmembrane proteins. That means the receptor protein doesn't pass back and forth through the plasma membrane several times. It just goes through one time. It's a single-pass receptor. But other types of tyrosine kinase receptors, like insulin receptors and insulin-like growth factor receptors, have multiple subunits. So it's not just a single pass. For instance, the insulin receptor has 2 alpha subunits and 2 beta subunits. Those subunits are all held together by disulfide bonds, which are very strong bonds. If you need to hold something together very tightly, you're going to use a disulfide bond. We often find disulfide bonds outside the cell in the extracellular environment where conditions are harsh, so you need a really strong bond to hold something together. So a molecule of insulin binds to the 2 alpha subunits, which are on the outside of the cell, and the 2 beta subunits that have a tyrosine kinase activity are actually found inside the cell. And when insulin binds to the alpha subunits, that causes a conformational change in the receptor, which allows the beta subunits to autophosphorylate. And that in turn allows the tyrosine kinase domain to start phosphorylating other proteins to have some downstream effect. That's it for tyrosine kinases, but let's quickly review some basic enzyme terminology, which is going to come up again and again. Again, an enzyme that uses ATP to add phosphate groups to something is called a kinase. An enzyme that removes a phosphate group is called a phosphatase. An enzyme that adds inorganic phosphate to something without using ATP is a phosphorylase. An enzyme that adds a carboxyl group is a carboxylase. And this usually requires biotin or vitamin B7 as a cofactor. An enzyme that removes a carboxyl group is a decarboxylase. Likewise, an enzyme that adds a hydroxyl group is a hydroxylase, and the enzyme that removes a hydroxyl group is a dehydroxylase. But I don't want you to get dehydroxylase confused with dehydrogenase. A dehydrogenase is an enzyme that oxidizes something by using an electron acceptor, such as NAD or NADP. So be sure you're familiar with all that terminology. One key function of the plasma membrane is to keep potassium inside the cell and to keep sodium outside the cell. Our cells use this sodium gradient to drive the active transport of a lot of different substances into the cell. Sodium naturally wants to enter the cell going down its concentration gradient, and the cell can use that energy to drive the transport of other things against their concentration gradients. So it's important for cells to be able to constantly fine-tune the sodium content within the cell. And potassium is also important because high potassium content inside the cell keeps the cell's resting membrane potential around minus 55, which is important for neurotransmission and muscle function and so on. So, how do cells manage to maintain the sodium and potassium gradients? You know this. It's the sodium-potassium ATPase. This pushes out 3 sodium ions for every 2 potassium ions that come in. And because sodium is constantly trickling back in and potassium is constantly trickling out, this sodium-potassium ATPase has to work continuously. And because it's an ATPase, you know that this process requires energy in order to maintain this gradient. You also need to know that the drug digoxin, or digitalis, is a cardiac glycoside that works by inhibiting the sodium-potassium ATPase. When we get to the cardiovascular section, we'll explain how inhibiting this enzyme will eventually increase intracellular calcium, and that makes the heart beat a little harder. Not everything that enters the cell enters through a protein channel. Lots of macromolecules enter the cell through endocytosis, which also requires some specialized equipment. So let's talk about receptor-mediated endocytosis for a minute. The molecule I want you to focus on is clathrin. Clathrin is a vesicular trafficking protein, similar to the coat proteins that we discussed with the Golgi apparatus. And clathrin helps the plasma membrane form spherical vesicles during endocytosis. Let's take a look at your study guide. Instead of binding to the endoplasmic reticulum or the Golgi apparatus like the coat proteins, clathrin binds to a protein called adaptin on the inner surface of the plasma membrane, and that adaptin is bound to a receptor protein. That receptor is going to bind some specific cargo that needs to be transported into the cell by endocytosis. It might be an LDL particle or transferrin or any number of things. Once that cargo molecule binds to the receptor, the clathrin molecules start to polymerize with other clathrin molecules. And just like we saw with the coat proteins, clathrin polymerizes into little spheres. So this allows the plasma membrane to fold up into a little vesicle. Then another protein called dynamin pinches that off and helps the two ends of the membrane fuse. Then you have formed a little vesicle that's coated with clathrin. And finally, the clathrin and adaptin can come off the vesicle and be reused, so you're left with a naked transport vesicle with just receptors and the cargo molecules inside. Then something needs to happen to the contents of that vesicle before they can be released inside the cell. Let's look at the next drawing. What typically happens after the uncoating is that the receptors get recycled back to the plasma membrane, and what's left of the vesicle fuses with a lysosome so that all those hydrolytic enzymes inside the lysosome can break down whatever was brought into the cell. Now in this example, it's an LDL particle, so the lysosomal enzymes are degrading the lipoproteins, and then the cholesterol is released into the cytoplasm. So this is the process of receptor-mediated endocytosis. Now let's look at this bottom image as an example of how this is clinically relevant. On figure A, you have the normal LDL receptor and adaptin and clathrin. And you can see that these come together in what we call clathrin-coated pits. But on figure B, there's a mutant LDL receptor that doesn't associate with adaptin and clathrin like it's supposed to. So these individuals can't bring the LDL into the cell and use it. So this is what you might see with familial hypercholesterolemia, where there's a defective LDL receptor. But we're going to talk more about that disease in another video. So that's clathrin. It's definitely an important mechanism to know about. Finally, let's talk about arachidonic acid derivatives, which are ultimately derived from plasma membrane phospholipids. Now, this is very important for you to know for your test, and it's very clinically relevant to know about how certain drugs interact with this arachidonic acid pathway. This is a 4-star topic, so don't breeze through this. Fundamentally, arachidonic acid derivatives are substances that allow for tissue-to-tissue communication. Things like leukotrienes and prostacyclin and prostaglandins are all communicating substances, and they're all derived from arachidonic acid. So let's look at this pathway in your study guide, and you can fill it out as we go along. You start out with cell membrane phospholipids, specifically phosphatidylinositol. Now, phosphatidylinositol is converted to arachidonic acid, which is letter A. Enzyme B is the enzyme that catalyzes this reaction. So B is phospholipase A2. That's an important enzyme to know. Phospholipase A2 is inhibited by corticosteroids, which is letter C. Now, corticosteroids have a lot of functions in the body. And one of the things they do is inhibit this enzyme, and so they inhibit the production of arachidonic acid. Then arachidonic acid can go down one of two pathways. First, arachidonic acid can be acted upon by letter D in the diagram, which is lipoxygenase, which forms hydroperoxides, which is letter F. And there's a drug that inhibits lipoxygenase, which is xylutone, which is letter E. Xylutone is a drug that's occasionally used to treat asthma because it blocks the formation of leukotrienes. So hydroperoxides are converted to leukotrienes, which is letter G. And that's the important thing to know here. There's leukotriene B4, which we're going to mention again in the immunology section, but it's associated with neutrophil chemotaxis. So if you want to get neutrophils to assemble in an area, you're going to try to break down arachidonic acid to leukotriene B4. And that's going to tell all the neutrophils, hey, come over here. And then other leukotrienes are involved with increasing bronchial tone, which is why blocking leukotrienes is helpful in asthma patients. So letter H is neutrophil chemotaxis and increased bronchial tone. Then letter I is two more drugs, zafirlukast and montelukast. These both have leu in the name because they block leukotriene receptors. And again, they interrupt that increased bronchial tone. So these drugs are useful in the treatment of asthma. So xylutone inhibits the formation of leukotrienes, and zafirlukast and montelukast block the leukotriene receptors. And you may want to know that montelukast is approved for younger age groups. So if you have, say, a 2-year-old with asthma that you're going to start on a leukotriene inhibitor, you should choose montelukast. That's something you want to know for your test. We're going to talk about that again in the pulmonary section. Now let's go back up to arachidonic acid, letter A, and look at the other side of the pathway. Arachidonic acid can be acted upon by letter J, which is the enzyme cyclooxygenase, either COX-1 or COX-2. Then letter K is a whole bunch of drugs that inhibit cyclooxygenase, including NSAIDs, acetaminophen, celecoxib, which is a COX-2 inhibitor, and aspirin. Now, aspirin irreversibly inhibits cyclooxygenase. And it's worth mentioning here that although acetaminophen inhibits cyclooxygenase, it's inactivated peripherally, so it has no antiplatelet effects or anti-inflammatory effects. Then letter L is endoperoxides, which are the products of cyclooxygenase. And then from there, the endoperoxides can go down 3 different pathways. Letter M is prostacyclin, or PGI2, which stands for prostaglandin I2. It may help you to think of PGI as being platelet gathering inhibitor because PGI2 decreases platelet aggregation. It also decreases vascular tone and bronchial tone and uterine tone. So it's going to abolish uterine contractions. So box N is decreased platelet aggregation, decreased vascular tone, decreased bronchial tone, and decreased uterine tone. And as a vasodilator, analogs of prostacyclin are used to treat conditions like Raynaud phenomenon and pulmonary hypertension. Letter O is prostaglandins, which are slightly different. Prostaglandins actually increase uterine tone, so they're helpful to either induce labor or they're useful as abortifacients because they induce uterine contractions. And they decrease vascular tone. Prostaglandins keep vessels open. We're going to say that again and again throughout these videos. Prostaglandins keep vessels open. Specifically, we're referring to prostaglandins E2 and F2 alpha. Prostaglandins also decrease bronchial tone, generally speaking. It's a little more complicated than that, but don't get bogged down in the details. And prostaglandins also increase gastric mucus production. So prostaglandins will protect the gastric mucosa in part because of this increase in gastric mucus production. So again, box P is increased uterine tone, decreased vascular tone, decreased bronchial tone, and increased gastric mucus production. And then the final pathway for arachidonic acid is to convert it to thromboxane, which is letter Q. Thromboxane is another important substance. Thromboxane is prothrombotic, as you might have guessed from the name, so it increases platelet aggregation. It also increases vascular tone and it increases bronchial tone. So that's box R. Increased platelet aggregation, increased vascular tone, and increased bronchial tone. It's kind of the opposite of what we saw PGI2 doing. So it's helpful to think about thromboxane and PGI2 as having opposite effects. So again, you should definitely spend some time with this pathway. Make sure you really know this inside and out, because you will see these substances again throughout the course and on your test. So that's it for the plasma membrane. Go ahead and work through the end of session quiz, and then we'll go over the answers together. All right, first question. Which arachidonic acid product causes each of the following effects? So increased bronchial tone is leukotrienes and thromboxane. Decreased bronchial tone is prostacyclin and some of the prostaglandins. Increased platelet aggregation is thromboxane. Decreased platelet aggregation is prostacyclin. Increased uterine tone is prostaglandins E2 and F2 alpha. Decreased uterine tone is prostacyclin. Increased vascular tone is thromboxane. Decreased vascular tone is prostaglandins, including prostacyclin. Next, what are the two most abundant substances in the plasma membranes? Well, it's proteins and phospholipids. And the last question, what drugs act on the arachidonic acid product pathway? And what enzymes do they affect? So corticosteroids inhibit phospholipase A2. Xyluton inhibits leukotriene production by inhibiting the enzyme lipoxygenase. Zafirlukast and montelukast inhibit the leukotriene receptors. And then the enzyme cyclooxygenase is inhibited by NSAIDs, acetaminophen, COX-2 inhibitors, and aspirin. All right, that's it for now. I'll see you next time.